talking about your graphs, you're going to have two kinds of data. You're going to have continuous data. There we go. The brain is a little slow this morning. And then discontinuous data. And you say, what's the difference? Continuous data is if you're tracking the mass, or you're tracking the height, or some other data point where it's the same specimen, the same organism, and you're coming back and collecting the same data point every day, okay? Discontinuous data is if you're collecting the same data point, but from different populations, okay? So some of you are counting fungal colonies, or you're counting the area of the zone, or actually the diameter of the zone of inhibition, okay? So you count 10 colonies over on this piece of flesh, right? And you count 8 colonies on this piece of flesh. That's discontinuous data, okay? So this would be from different populations. And then how you graph this data is also very different. If it's continuous data, it's a line graph. If it's discontinuous, it's a bar graph. Okay. You're going to have two sets of graphs. Okay? You're going to have your first set of graphs or figures, right? That are your raw data. Okay. The height, the number of colonies, the, what did I say, the diameter of the zone of inhibition. That's your original data collection, okay? Then you're going to have the second set of graphs, and those are going to be your averages, your rates. So this is first set. And this is your raw data, okay? And then as soon as you start doing calculations on that data, that's a second set of graphs, okay? These are going to be things like growth rate, or if you're calculating area, or if you're calculating average, or if you're doing amount of change, like the difference between populations. Okay, these are all calculations, right? All of these are bar graphs, all of them. Okay, so your continuous data, mass, height. Okay, let's say that you're growing a plant, and so you collect it every day, or every other day, or once a week, whatever you decided to collect in your protocol. Which reminds me, I'm digging into your protocols today. That's my plan for after lunch. I'm hopefully going to do them today and have them done by tomorrow. That's my goal. We'll see. So you then collect, let's see, the height of the plant here, all right? But then once you take the height of the plant divided by time, that gives you rate, and then rate is then your bars, okay? So you're going to have two sets of graphs, okay? Your raw data, then your calculations, right? And I would color coordinate all of this, okay? Because you get into, this is when the color gets really super cool. So if this is population 1, this is population 2, and this is population 3, and I don't know, let's see, this is average height. Okay, whatever colors you choose, oh, let's see, let's go a little blue. And then let's go, what is this, pink? Sure, it's pink. That's a little light. Let's get that darker for you. There we go. Let's do that. And then, oh, maybe a purple. That'll be dramatic. There you go. Okay. So if you're setting up the average height of your plants, okay, whichever colors that you choose here for your populations, once you choose the color here, what we're going to do is we're going to take this color and we're going to go back to your data collection. Okay. You don't establish your colors until you've done your data analysis. You then have your data collection, and in your table, such. in your table, you're going to have population 1, population 2, and population 3. Okay, whichever colors you chose here in your data analysis, we then go back to your data collection, and I misplaced my blue, there it is, and then you bring these same colors into here, just like this. It's a wonderful way to help the grader visually tie all your data together. So that way they see it all. Okay. And then, Antonio, when you were talking about putting your 3 trials, okay, if you wanted to color coordinate your trials, that would be awesome too. Okay. But we can't color coordinate your data collection until you make the colors in your data analysis. Okay. That's why I didn't tell you about the coloring your tables, because you don't know what colors you're going to choose yet. You have to get into your analysis, and then that folds back to your collection. Any questions on that? You guys are cool?
Yep.
Okay.